Greetings. Today, I'd like to speak about introduction to infectious diseases data as part of MMED course. These slides have been made by myself, but also previously by my colleagues, Faika, Steve Bellan, and Julie Pulliam. For today, the key things I want us to understand are definitions of infection, what is disease, what are infectious diseases. We understand the approaches to collection of infection data and disease data, and also we'll talk about the metrics to measure presence and spread of infectious diseases. I think by the end of this lecture, you should be quite clear about the differences between infection, disease, and infectious diseases. So these are interesting concepts in themselves. What is infection? What is disease? And what's an infectious disease? So if there was an opportunity to sit and ask this question, I would ask, what would you define as infection? In this case, I think we have really good examples from the recent past. Right now, we are in the midst of the COVID-19 pandemic. Is COVID-19 an infection or is it a disease? I mean, you could think about it in terms of when we define infection, we are implying that there is one organism, which could be a parasite, or it could be a pathogen that has entered another organism. In the current case of COVID-19, we are referring to coronavirus being in the host, and the host in this case is a human being. So coronavirus is what causes COVID-19, the disease. So that's really important to know, because in terms of how we determine how to control diseases or how to control infections, that concept is really important, and we'll see that before the end of the lecture today. If you think about diseases is very much a departure from the normal, working of a body or working of an organism. That we refer to disease, especially if there is an effect on its survival or probability of surviving, or even its productivity or reproduction. So once you've got a deviation from the normal physiological status of an organism, we refer to you as having a disease. Not every disease could be out of an organism or an infection. Some diseases could just be disorders that are genetic, but others could be connected to a pathogen. And that's the main interest within this series of lectures that we have at MMED. We're dealing with infectious diseases and how those manifest in hosts or in people or in animals, and how that can be controlled at a population level. So for us to do this kind of thing, we need to be clear about the differences between disease and infection. Let me take the example of some work we have done previously. Imagine a calf born of a cow that has been born and has to spend the first year of life. It will experience many things that might not allow it to see its first year birthday. Many of those could be infections, others could be accidents. And here's a good example of some work where we have looked at calves in their first year of life and trying to understand what kills those animals, before they can reach their first year of life. And here what you've done is we have investigated, by getting over 500 calves and following them at least every month, collecting all information about their clinical experiences, drawing blood and any other sample out of the animal, and finding out if they have got certain pathogens or parasites or infections. And what you get out of this comprehensive data set is a mix of many reasons why animals die in their first year of life. Here we have a mix of infectious diseases like East Coast fever, and also we have got other causes of death that are not infections. For instance, what we refer to as mismothering. The mother of the calf or the cow was not able to take care of and provide enough milk, and therefore the animal did not survive the first year. But importantly, it's good to see that even though we have all this nice clinical information about these animals in their first year of life, for 25 of those 88 animals, we couldn't tell what killed them. You couldn't lay your finger on the infection that the animal had suffered that could have resulted in death. And that's important because when you're understanding, when you're seeking to understand what happens to organisms, you should know that you don't have all the answers yet. Even when you have got really good diagnostic kits and diagnostic tests, you don't always arrive at a clear diagnosis of what is causing disease or what is causing death. I mean, good examples of infectious disease. I mean, in the recent past, we have had really major ones that the world has been aware of. So, for instance, the current pandemic, the infectious disease agent for that is a coronavirus. It's a virus. Previously, we were dealing with the Ebola epidemic in West Africa, and we try and use the Ebola epidemic in West Africa as a good example for us to understand infectious disease data, or even for infections that like HIV, a virus, or tuberculosis, which is caused by a bacteria. And these are just microorganisms that are living their lives, but living their lives within a host or within a body, a human, an animal. And where they create certain pathological disorders that manifest themselves in form of a disease. So you could think about what are the causative agents for infectious diseases. They could be viruses, they could be bacteria, they could be fungi, and all those, when they cause, important disorders within the body, then we refer to that as. But you should also remember that there are many infectious agents that even when you have them in our bodies, they do not manifest in disease. So they could be harmless, and some of them could be very harmful. And that is important to note. What's the impact of each of these different infectious agents coming into a host or into a body? So the work of infectious disease epidemiologists, or what you'd sort of refer to as disease detectives, is to understand patterns of disease, not just at an individual level, but at a population level. If you think it's like having a forest, a clinician will see a patient at a time, a public health specialist or an epidemiologist will sort of think of a group of patients and having certain characteristics. So the idea for this disease detective work is to unravel these patterns of disease that are happening at population level. At a host level, you also care for how does an individual get exposed? When do they get infected? How does the infection progress to disease? What are the outcomes of that disease? The idea of this, being an epidemiologist is to not only understand the patterns of disease, but also be able to associate it with certain factors that either increase the risk of disease or decrease the risk of disease. And importantly, come up with what are the control options available to minimize the impact of these infectious disease agents. And so you could think about moving from infection to disease from the time, somebody gets the pathogen or the microorganism into their body to when it starts manifesting in form of either clinical signs or in form of shedding itself, moving on from one host to another. And to understand that, we try and simplify that into periods. So you'll hear often people talk about incubation period, you hear about a latent period, you hear about the infectious period. And all these are really important when you're thinking about the control of an infectious disease. And you could try and make read a little bit more about these differences, but I want to just spend a few minutes telling you, or at least demonstrating to you, what are the differences between these three different periods. Yeah. So take the example of infection to disease, the continuum. You get infected, or the microorganisms get into the body. For a period of time, you will not know. And often we know that you have been infected once we start feeling unwell and start having clinical signs. So from the period when you get infected with the parasite to the period when your symptoms or your clinical signs start showing, that period is referred to as the incubation period. Now, you also can get infected and start passing on the pathogen, or shedding the virus, or shedding the bacteria before your clinical signs have shown or have been demonstrated. And that period, when from the time you get infected to the time you start being infectious or contagious, that period of time is referred to as the latent period. And it does not necessarily mean that incubation period is equal to latent period, because you can be infectious ahead of having clinical signs, or you could be infectious even in the absence of clinical signs. And I think a perfect example is what you've dealt with the pandemic, the COVID-19 pandemic. We know that there's a group of people who can still pass on the virus but don't have any clinical signs. We have referred to them as asymptomatics. And there are people that, even though you're showing clinical signs, you possibly are not shedding, because the period of time when you are contagious or infectious is not equal to when you have clinical signs. So you could be still having, you're struggling with breathing, or you're struggling with fever, but you are not shedding that time, because the infectious period is not necessarily equal to when you have clinical signs. All that sort of clarifies it quite clearly. And you can think of, how would we then measure infectiousness? How do we know whether somebody will pass on the infection? There are proxy measures of this. So to give the example of the current pandemic, COVID-19, one way is to do a PCR test, or do this diagnostic test that look for the virus. So either an antigen test, what they refer to as an antigen test, or what they refer to as the PCR tests. So if we can identify the pathogen in you, and it's likely that you are passing the infection. So if we identify it, say, from a swab, then we tell that if you are coughing or shedding saliva, then that will contain the virus as well. The other way to do it could be through experiments or observational studies, and this is done often, particularly within the animal studies, where you can demonstrate transmission of a pathogen between individuals by having a sick individual and having a healthy individual, and seeing whether this healthy individual will get the disease through just contacts with this individual that's unwell. Naturally, that kind of thing is done. Say if somebody tests positive at a household, do you see other members of the household getting the infection, and how long does that take? So that's some nice proxy ways of measuring movement of pathogens from one host to another, either by testing fluids within the body, or being able to demonstrate transmission of pathogen between individuals. So there are a number of those parameters I'm mentioning here are really critical for making decisions on day-to-day life. So for instance, if you think about measles, or you think about seasonal influenza, you want to estimate what is the incubation time, you want to know what is the latent period, you want to know for how long is someone infectious, and for how long do you expect clinical signs to go on? And that's important because imagine of an infection that is passed, like between children. An example is measles or seasonal influenza. You then have to make a decision at what point does a child, when would you require a child not to go to school? Because if they went to school, they would possibly infect other children. At what time would you say they require before they can come back to school and they're possibly safe? And I think you've seen that a little bit during the pandemic, like if you go into quarantine or if you go into isolation, for how long do we keep you in isolation, or how long do we recommend that you need to be isolated before you can mix freely with other members of the public? So understanding the period of infectiousness is really critical for deciding how to control infectious diseases, particularly within outbreaks. Let me take the example of, and I'll mix animal diseases and human diseases, sort of to put the case forward that, the methods that we use for studying diseases in animals and humans are not very different. So here is a really nice example of a study that tries to estimate how much time it takes, for foot and mouth, which is one of the big cattle diseases, to incubate. What's the latent period? What's the infectious period for foot and mouth disease in cattle, which is passed really through contacts and close contacts between animals that are exposed, that are infectious and those that are susceptible. What do you think is really important to define in this kind of study? One of the key things is the time of exposure. When do these animals get exposed? The second one is, what are the clinical signs that are indicative of a foot and mouth disease episode? And so in this study here, what they have done is they have used experimental data by placing animals that have been infected and animals that are susceptible, and then observing how long does it take before, and getting samples really frequently to determine at what point do animals And the key answers they get from this is that they found there were shorter latent periods than they were imagining from observational studies previously, and much longer, estimates of the mean infectious period, how long an animal remains infectious, by up to 4 days. And the key take home of this kind of study was animals were not infectious until about half a day after the onset of clinical signs, which I think is a really good finding, saying that the period of infectiousness starts after clinical signs have been seen, which means in terms of the control of this disease at a population level, you could use clinical signs to minimize, as a surveillance tool to minimize when the likelihood of the disease being transmitted. And that means that they refer to that like preemptive versus reactive control measures. So I think it's just a nice piece of work that if I can tell you have clinical signs, and after clinical signs, that's when I start becoming infectious. I can look at any farm that has an animal that shows the clinical signs, and I know I can actually just put restrictive measures immediately, and that disease is unlikely to have left that farm to another farm. But one of the really key important things is to define a case. Whenever you're faced with an infectious disease episode, the first thing you want to is to say, what is a case? Which essentially is a set of standard criteria that you're using to decide whether a person or an animal has a particular disease. And what you think of is you go to define it with who, where, when, what. Basically, we refer to that as people, place, time, and then you have a clinical description of that. to describe a case. To give the example again of the foot and mouth disease, which is a cattle disease, and looking at a big epidemic in the UK that happened just about 20 years ago. They first see the first case, around 19th of February of the year 2001, and they note that when they take the pigs to the slaughterhouse in Essex, they show clinical signs of foot and mouth disease, foot and mouth disease. And then immediately, 2 days later, because it's a big thing, a country that does not have a disease tries to keep itself free of a disease. If there is an incursion or if there is a case of that disease, then they want to do the best they can to get back to freedom from that disease. And so there's a series of events that follow up. There is a ban on all British exports of livestock meat and animal products. Then another case is found in a different place, and another case in Cornwall, within a period of about 5 days. And the number of cases within a month of finding the first case are over 200. So how would you define a case of FMD? I think it sort of depends on where you are at in the pandemic. And take this good example here. We are looking at the number of cases that began in Feb of 19th of Feb, and you can see they go up and go down, and you have big measures that have been marked by these nice red lines here, showing the movement restrictions when there was a welfare call, when they decided that for every premise, we're going to look at 3 kilometers around it and can stop any movement and cull all the animals in there, to when you really end up with very few cases. The thing to note about this is that whereas I have so far talked about a case being at an individual, either an animal has this kind of clinical signs or a person has this kind of clinical signs, you could have a case definition that is more than one animal, more than one host. So you can go beyond an individual animal into a herd. So for instance, the description of foot and mouth disease in the context of the FMD outbreak of 2001 was very much herds. Like if a single animal showed the clinical signs, or there was a demonstration of the foot and mouth disease virus, then that herd was condemned as a whole, or at least it was referred to as a case. So a case is not necessarily a single animal. It could mean that there's no way of distinguishing between the animal that you sampled and whether these other animals are negative. And so you take the whole herd to be infected. If you think about it, like in the context of, say, the pandemic, we know about individuals, but sometimes if you have like a new variant of a virus, countries can make decisions about whether flights could leave country A to B based on whether they have seen the variant. Meaning the case definition in this case is not the individual that has the variant of concern. It could be a group of people that are likely to have contacted that kind of person. And so you sort of think about how do we control or reduce the risk of the infection moving from one country to another. Well, let's take the example of, thinking about infection over time and over different places. Here is, an example of the big Ebola epidemic that happened in West Africa. And you're looking at 3 countries here: Liberia, Sierra Leone, and Guinea. And what you're measuring here, if you see, is very much cases per week. So we are not just looking at one individual, you're looking at every week and getting the number of cases that we see in that week and lumping that together and are able to follow over time. And you can see from like this nice graphic here that it starts a little bit earlier within Guinea, and then after that you have a few cases in Liberia, but you have a big epidemic happening around Liberia around this time, somewhere in August and November, and also in Guinea as well. And you have many other differences that you can see within the countries. And so in talking about case definition, think about this nice case study of how Ebola was spreading within West Africa. It begins in a little village at the border of the Ghanaian village of Meludu, where you sort of noted a boy who began suffering from a fever, headache, and had bloody diarrhea. And over the next 4 months, his immediate family and all the contacts and close community members begin to die of the disease. And when treating these cases, healthcare workers and doctors believe that they were at the moment dealing with a disease like cholera or Lassa fever, which are much less deadly compared to the virus that was later to be known that this was actually an epidemic of Ebola. And at the beginning, this was never referred to as an Ebola outbreak, and it was only, referred later once the people knew that this is not the usual cholera or Lassa fever that you have seen before. And so the sort of an evolution of case definition of the EVD, Ebola virus disease within the region. So it's, again, if I refer back to what I was talking on before, you have a set of standard criteria for deciding whether a person has a particular disease or not. And in this case, at the very early stages of the pandemic, of the epidemic within West Africa, the person here was the resident of this village that where the boy was first diagnosed with Ebola virus. The place is West Africa. So if you lived in East Africa like myself, at this point, somewhere November 2013, even if I saw a case that sort of showed elevated body temperature, fever, severe headache, that would not be a suspect case of Ebola, because Ebola at that point is only referred to as happening within this very small village in West Africa. But obviously, over the time period, as we follow the outbreak, there was clear evidence that this appeared to be seen in more places than that initial village. And when investigating those outbreak of Ebola, investigators sort of go back and look at the number of cases that have been noted over time and can decide how far the disease has spread. So sort of if you look at when the outbreak begins, it's just in a few areas here where it starts. And then, as you move on towards early 2014, mid of 2014, we are seeing more cases within Guinea, within Liberia, within Sierra Leone, and a year down the line, then you have a lot of cases being seen. That is measured as average new cases of each week. And so when you're investigating this Ebola epidemic, you sort of begin with this small, very restricted case definition, but as you learn more and learn that there are many more places that the disease is happening, then part of your people, place, and time changes. So places, not just the Guinea where it started, they start moving to Liberia and even other places. So, sort of, if you remember Jonathan's lecture, the first lecture on public health and epidemiology, you could think of what you see if you were to wait for cases at the health facilities or at hospitals, and what is happening, in the community. So you could sort of think about like clinical epidemiology, where you're looking at patients who show up at a health facility, and population-based epidemiology, where you're thinking about how much of the infection is happening at our population level. And you have sort of the people who end up at health facilities is really a tip of the iceberg. The disease or the infection could actually be much more larger within the community than you see it if you only are waiting for it within the health facilities. Okay, so I think the key here is you are interested with the new cases that are confirmed or probable cases that are happening over time. And if you now think about how did this move on to the other parts of the world, by the 10th of October of 2014, WHO had reported different kinds of cases that were found around the world. So there were counts of cases, the counts of death, and also the report, not just not just the count of cases and deaths, but also even suspected cases. And this is because the disease, people move into new places like the United States. You have, you don't know whether cases have gone to Brazil, and so you get a definition of not just confirmed case. You have a definition of which are those that are probable cases, which are those that are possible cases, which of those could be suspect cases, and you also need to have a way of determining that this is really not the case, it's not a case. And so in defining how, case definitions, we go with 4 or 5 main ways during an epidemic. So you have a confirmed case, and a confirmed case in this case is you have the clinical signs, you meet the people, place, time, but also you have got a laboratory confirmation. In this case, maybe take the example of the current pandemic. It means you have come into contact with a person who has had corona before, COVID-19, or is suffering COVID and is showing the disease clinical signs at the moment. But also, once we take a sample from you, we can confirm the presence of the virus. So laboratory confirmation, that would be referred to as a confirmed case. The other way to categorize a case would be a probable case that has everything, signs and symptoms in an individual that meets the person, place, time criteria, but also you have not confirmed with a lab specimen, but you know that this person has been into contact with a person with more specific clinical signs, and that would be referred to as a probable case. A possible case here in this case would be, again, signs and symptoms and individual meeting, the person, place, time, and criteria, plus maybe a physician or like a healthcare worker making a suspicion that this could be the case of interest. So adding physician diagnosis to make that a possible case. A suspect case is, signs, symptoms, and individual meeting, place, time, and criteria, and possibly don't have a physician diagnosis or don't have a known contact with more specific clinical signs or even a lab confirmation. And not a case is when you fail to fulfill the criteria for either confirmed, probable, possible, or suspect case. Sometimes people say it's not a case also because you have tested and you cannot see the causative agent of the disease. So just to recap, when you talk about case definition, this set of criteria for deciding whether a person has a particular disease or infection, remember, changes over time, and that is informed by what is happening within the pandemic or within the disease outbreak scenarios. So we began, for instance, with Ebola, has residents of Melidu, recent visitors, West Africa, Guinea as a place, time on or after. So you sort of don't say that's a potential case if it happened 3 years down before when you're not expecting it. And then you have a series of clinical descriptions that can help define the case. I think the one thing to note about a lot of these infectious diseases that it's sometimes the clinical signs can be common across many infectious diseases, and therefore it is not just a clinical description, it is a clinical description plus epidemiological intelligence of what we are referring to as people, place, and time included as part of it. Okay. So over time, the case definition changes. So we move from just residents of that one village and we go into recent visitors to West Africa, people who have been in Senegal, Guinea, Sierra Leone, Liberia, as well as their close contacts or others in the community. And you remember around the Ebola epidemic, a lot of countries made decisions as to not based on whether they had tested you and found you with Ebola virus disease. It's also if you had visited those countries, and they would say, if you have come from any of these countries, we will not let you in. And I think that you've sort of seen that quite common as well with COVID-19. And when the disease starts spreading across, then the place is no longer important, or it's worldwide. So it's like the case we have now with the COVID-19 pandemic. Initially, we started with Wuhan, then we went into different places in China, went into Europe, Africa for a period of time, did not have a case, but once it was declared a pandemic, we started seeing the cases all over the world. So now if you meet, the case definition, place means just anywhere, because the disease is everywhere in the world. Until that time that you have been able to control it, then it means quite a high suspect case. And then the definition of the clinical descriptions, I think you have seen even the changes in the clinical descriptions of say COVID-19. Initially, it was very much a respiratory infection or symptoms that were connected to the respiratory system. But after some time, people started talking about even gastrointestinal diarrhea as also potentially being a way of demonstrating infection. And then we discovered that only a small proportion would have clinical signs. So being infectious is not necessarily connected to whether or not you have clinical signs. And that's really important to show that even though we have case definition is an important component of understanding infectious disease data, that case definition is not static and it keeps moving with time. Okay, so we have sort of spent some time talking about defining a case. I'd like to spend just a few more minutes talking about the different approaches to data collection and presentation. And I start with really a disease that I work on, which is rabies. Rabies is transmitted by domestic dogs mainly to humans. It's a virus. And more often than not, if you have clinical signs of rabies as a human, you most certainly will end up in death. So it's one of the highly fatal infectious diseases. So here we got some nice data from Tanzania, collected by Kerry Hampson from the University of Glasgow, looking at two districts. We have Serengeti and Gorogoro. And here they have collected data. Number of cases, which are blue, is what you see in Serengeti, and red is what you see in Gorogoro, for a period between 2002 to 2007. And it's data on rabies transmission in these two places. And you're able to monitor the spread of the infection using a really nice method that has now become quite common during the pandemic. And this is what we refer to as a contact tracing method. So you have a case and then you go actively looking for more cases that might be associated with the first case that you saw. And that's an example of how you collect data. So you're doing it with surveillance, you have contact tracing or outbreak investigations. So if you think about it, and I think now it's pretty easy to understand given the COVID-19 pandemic you've been experiencing, what are the ways of collecting data on cases? You could do it as a passive way, or you could do it as active way. If you think about passive ways of collecting infectious disease data, is like you are a clinician, you wait for people from the community to come to the hospital, and then you know how many cases are coming from the community. That's passive. You're waiting for It's waiting for patient to come to hospital. The other way is to do active surveillance for infectious disease. And this means that you get a case, but go search actively for other cases. So for instance, the common method now we know about active case finding is the contact tracing, where if you are infected, say with SARS-CoV-2 that causes COVID-19, a lot of places you have contact tracing where they go and look for all contacts of infected persons and try and isolate them. So it's you test and then you search and you trace and then you isolate. Test, trace, and isolate as one of the key approaches to minimizing the spread of COVID-19 that has been employed. And the other way is to carry out what we refer to as epidemiological studies. So you could say, I have a case, several cases coming in, and and I'm studying them, and that would be like a case series. Or you could say, I want to understand what causes disease or risk factors for disease, and you have a case, and then you compare with people with almost similar conditions but don't have the case, to identify what might be that differentiating factor for why one has a case and one does not have a case, or the outcome of this infection differs from the outcome of not having an infection. Or other ways is to say have a cohort where you have a group of animals or group of people, and then you can follow them over time to see the differences in the risk of getting infected and not getting infected. Or in response to events like the outbreak investigations that we have been conducting now around COVID-19 in many parts of our countries. So moving away from the active and passive data collection, you can think of metrics to describe the spread of disease, which is very much related to what we'll be doing here at MMED. So take again the example of Ebola in West Africa. What I'm showing here is the whole of West Africa and also individual places, Guinea, Sierra Leone, Liberia. We are looking at the number of cases that are seen over time, and this tells you we have an increasing epidemic here, that you're seeing more and more cases, particularly from May 25th moving on to September 14th, for which the data is being shown here. But so if you think about how do we measure disease within a population, one of the really key things is finding new cases. And that's important, finding new cases, because new cases just will tell you how quickly, for instance, a disease is spreading within a population, as opposed to just looking at like cross-section. We talk about that in a minute. So you could think of incidence of infection. An incidence of infection here is you're moving from when you get infected to when you can see onset of symptoms, or when you get infected to onset of shedding. And everyone who gets infected or get exposed becomes infected. That's the assumption you're making right here, but it's not necessarily the truth of what happens, in normal times. Not everyone who gets exposed becomes infected, but here we are making an assumption that if you get exposed to a person that has infection X, that you get infected. So incidence of infection would be this part here, where you're moving from susceptible to exposed to infectious. This part means you are getting the infection, and that's the new cases would actually be falling here. So new cases, the number of people that would move from this susceptible group to exposed group, and I think you remember that from the previous lectures that we have been given around SEAR. So that part there would be the new cases that you're getting within a population. That would refer to the incidence of infection expressed in a mathematical way, particularly for modeling. How about prevalence? So if you think about what prevalence is that if I look at any one single time, how many people either have the infection or have previously had the infection? How common is that infection at a single time? That's what prevalence would be. In this case, it's really a combination of all the people who have been exposed and all the people who have been infected over the entire population, and that's what would give you prevalence in the context of modeling, mathematical modeling. You could think about, how do we measure prevalence? How do you know whether people have been exposed? One common way is to study what we refer within epidemiology as seroprevalence. So anytime an infectious agent goes into a host, a human or an animal, the animal will produce some signals that help fight the foreign body. And those signals will be referred to as antibodies. They are anti the infection. And the body sometimes keeps memory of that very well. So as even if I come 3 years down the line, I'll still get signatures of whether or not I have been exposed to this infection before by looking at these antibodies. So when you determine the prevalence of an infection, we often look at antibodies, the likelihood of having antibodies within the hosts that are specific to the infection, and that would be the seroprevalence of the infection. So to take the example of HIV, and here you're looking at in South Africa here, UNAID data ranging from 1986 or so to 2006. And you can see a comparison of HIV prevalence among pregnant women being shown in red, and also among sex workers being shown in little black dots. I think what you see here is that over a period of time, there are things you can draw conclusions from looking at this data. That first of all, the prevalence of HIV was going up and then seemed to level at some point. But importantly, that the sex workers have a higher prevalence compared to just, the population of pregnant women. Why would you be then interested in measuring, instead of prevalence, looking at incidence? In this case, the person that survives between 1990 and 2006, every time you test them whether they have had exposure, you see that because of those long-lived antibodies, you sort of see them that they have at one point been exposed. But if you wanted to study the dynamics of HIV infection in that region, incidence would actually be better, new cases, because new cases would tell you whether you still been able to control the disease or more and more people are getting infected, because prevalence, is complicated by the fact that we have new cases, but also very old cases of the same disease. So the mathematical expression of seroprevalence is what I've shown you before. In this case, you know that people who are recovered would also have been exposed, at least if you have evidence of presence of antibodies. So in this case, then R over N, which is the recovered over the number of people there, then that to sort of give you the prevalence. Okay, so I think the really critical thing to note here is that seroprevalence can be related to the prevalence of infection, but also past exposure, and that's important to always remember. The other thing to note is that it is not all the time that having antibodies means you're protected against the infection. So you could have antibodies but not be protected or not be resistant to the infection. And I think we're hearing a little bit of that with COVID-19, where there are questions as to, once you're exposed or vaccinated, how long does your immunity last? If I come down 2 years down the line and you're exposed to another virus, coronavirus, that causes COVID, would you still get the disease or would you be protected? So it's important to note that presence of antibodies does not always relate neatly well into protection or resistance. Okay, and as I come to an end, here's a really nice example of seroprevalence, of what we refer to immunoglobulins G antibodies against measles. And what you see is, among males and females, that if you're really young, you're born with very high percentage of IgG positive, and after some time that goes down, and with time that keeps going up to a point where almost 100% of the people have IgG. This tells you that this could be associated with maternal antibodies that are passed on to neonates and to children, and after some time that keeps waning, and then you start getting exposed to these infections, or you get vaccinated, and that keeps going up to a point where almost the whole population would have evidence of past exposure to either the vaccine or at least to the virus, to the measles virus. And you can think about, that nice graphic that's showing what's happening at population level, but it's aggregated data, but that data comes from individuals. So part of dealing with infectious disease data is also knowing data that is coming from individuals and how to aggregate it to speak about the population. And here's an example here where we are looking at each of these should be representing an individual, they identified. You can't tell which individual it is, but they have their life histories shown there. But also you could have aggregated data, in which case you're not looking at all these cases each individually, but you're taking the number of cases that happens between these weeks and between these weeks and between these weeks. And those levels of aggregation are really critical as you think about infectious disease models. Here's an example. Remember I told you about this, many curves that we are looking at the first year of life. And here is an example of the life histories that you have to deal with in terms of infectious disease data. So all these represent when curves have been recruited, and each of these dots represent an event. So like all the reds will show when animals died, all the blues will show when animals had clinical episodes. So what you try to do with infectious disease data is get these individual life histories and then find parameters that you can get out of this population that you can use to define your simplified representation of the complex biological world into mathematical equations that can help you determine how well you can stop a disease or control a disease. And another good example here is just an investigation line list of people who have been exposed to anthrax within Kenya. So you have got individual case one, you can tell about the symptoms, when the data was collected, whether they got hospitalized, if they survived, what date they were discharged. Unfortunately, if they died, when did they die? And then you can use this information to just understand how to describe the parameters of the disease within the population by use of these kind of things. But this is the identified data. Even though I have information at really individual level, I can't tell which individual this is. And that's for ethical reasons, that's how you need to have your data maintained. Okay, so you're going to go into a lab sometime soon where you learn how to work with infectious disease data, visualize this kind of data, and to say, well, we have perfect tools now in R that help make it easy to demonstrate this kind of thing. But just in case you get frustrated working with R, here's a nice graphic to remind you of the other graphic capabilities that might require if R became very difficult. At least you don't have to plot your data by hand. Great, many thanks. I hope you have enjoyed it.